السلام عليكم ورحمة الله وبركاته. الحمد لله رب العالمين، والصلاة والسلام على أشرف الأنبياء والمرسلين سيدنا محمد وعلى آله وصحبه أجمعين. This is a video recording for the first physiology lecture in the cardiovascular block. The main topic of this lecture is contractile mechanism in cardiac muscle. Inshallah, we will have a series of video recordings for all the physiology lectures in the cardiovascular block. These are the active learning objectives, the intended learning outcomes, the ILOs for this learning activity. The ILOs are statements which begin with active verbs like describe, explain, discuss, explain, summarize. These statements are important because they describe what is expected from you after you have reviewed the PowerPoint presentation and the associated learning resources. They should direct your learning and the level of the learning which you should have acquired after the successful completion of this learning activity. And that's why I always put the intended learning outcomes, the ILOs, at the beginning and also at the end of the PowerPoint presentations of the physiology lectures. Here we have the recommended learning resources for this lecture in the form of textbooks of the well-known physiology textbooks, Guyton and Hall, textbook of medical physiology, Linda Costanzo Physiology, and Gano's Review of Medical Physiology. The English language of Linda Costanzo Physiology book is very simple and the diagrams are excellent, and that's why I recommend that every one of you has a copy of this book. As you all know, the human body is composed of a number of body systems with coordinated and integrated functions. One of the important body systems is the cardiovascular system. This system consists of the heart, which functions as the central pump of the cardiovascular system, the blood vessels, and the blood which circulates along the blood vessels. As you can see in this diagram, the cardiovascular system is a closed system through which blood circulates, and that's why this system is also known as the circulatory system, or simply the circulation. The cardiovascular system plays important functions in the body along with other body systems in order to maintain the body in a good health. Some of these very important functions include the delivery of oxygen, glucose, and other nutrients to the active tissues, the removal of carbon dioxide, lactate, and other metabolic waste products, the byproducts of metabolism from the active tissues, and the participation in maintaining homeostasis in the body and maintaining the body in good health. Interestingly, the cardiovascular system not only delivers oxygen, glucose, and other nutrients to the active tissues, but it adjusts the supply of oxygen and nutrients to the tissues in different physiological states. And by being the major transport system in the body, the cardiovascular system transports metabolites and other substances to and from the storage sites, and it also transports hormones from the endocrine glands to their target organs and tissues, and it also transports antibodies and other substances to the site of action. The cardiovascular system also plays an important role in defense and in thermoregulation. Now, let's very briefly describe the functions of the major components of the cardiovascular system, and let's begin with the heart as the central pump of the cardiovascular system. The heart is a muscular organ composed of 4 chambers, 2 atria, and 2 ventricles. The 2 atria, the right atrium and the left atrium, are thin-walled, low-pressure chambers. They function as receptacles of blood and blood reservoirs for their respective ventricles. The right atrium functions as a blood receptacle and a reservoir of blood for the right ventricle, and similarly the left atrium functions as a blood receptacle and a reservoir for blood for the left ventricle. Under normal conditions, the atria are not important as pumps. The ventricles, the left and the right ventricles, are the principal pumps of the cardiovascular system. So in the heart, we actually have 2 pumps, the right ventricle or the right-sided pump, and the left ventricle or the left-sided pump. It is clear from this diagram that the right ventricle is considerably thinner than the left ventricle. In fact, the thickness of the left ventricle is 2 to 3 times as thick as the right ventricle. The thickness of the ventricular wall reflects the pressure which is generated by the ventricle. The left ventricle generates much higher pressure than the pressure generated by the right ventricle. The pressure generated by the left ventricle is 6 to 7 times higher than the pressure generated by the right ventricle. However, it is important to note that the flow which the left ventricle generates into the aorta in a minute is equivalent to the flow which the right ventricle generates into the pulmonary trunk. However, the flow which is generated by the left ventricle into the aorta is generated with much higher pressure than the flow generated by the right ventricle into the pulmonary trunk. The contraction of the cardiac chamber is physiologically referred to as systole, while its relaxation is termed diastole. A heartbeat consists of systole and diastole of all the 4 cardiac chambers, and the heartbeat is also physiologically referred to as the cardiac cycle. The atria and the ventricles undergo separate cycles of systole and diastole, and this will be described in details in 2 lectures on the cardiac cycle. In the heart, we have valves at the entrance points of the ventricles, between the atria and the ventricles. These valves are known as the atrioventricular valves, the AV valves. We have the tricuspid valve between the right atrium and the right ventricle, and the bicuspid valve or the mitral valve between the left atrium and the left ventricle. We also have valves at the exit points of the ventricles. These valves are known as the semilinar valves. We have the aortic valve between the left ventricle and the aorta, and the pulmonary valve between the right ventricle and the pulmonary trunk. The edges of the atrioventricular valves, the tricuspid valve and the mitral valve, are attached to ventricular papillary muscles by the chordae tendineae. This attachment of the edges of the atrioventricular valves to the ventricular papillary muscles by the chordae tendineae is important in order to prevent eversion of the valves during ventricular systole. In order for the heart to function as the central pump of the cardiovascular system, there are specific requirements for effective pumping of the heart. The first requirement is that the contractions of the individual cardiac muscle cells, the myocytes, must occur at regular intervals, and these contractions must be synchronized. They must be at the same time. The myocytes should contract at the same time. Their contractions must be synchronized. And the contraction shouldn't be arrhythmic. Another important requirement for effective pumping is that the cardiac valves must open fully. They should not be stenotic. We shouldn't have narrowing of the valvular orifices. And narrowing of the valvular orifices is known as stenosis. The valves must not leak. This is another requirement for effective pumping. The valves should not be insufficient and should not be regurgitant. The ventricular contractions must be forceful. We shouldn't have failure of either the left ventricle or the right ventricle. And equally important is that the ventricles must be adequately filled with blood during diastole. Here we have two questions. The first question is, specify 5 different mechanisms which can lead to ineffective cardiac pumping. The second question is, think of 5 possible cardiac diseases which may precipitate ineffective cardiac pumping. And I leave these questions for you to answer based on these 5 basic requirements for the heart to function as the central pump of the cardiovascular system. Now let's describe the functions of the blood vessels, and after we finish with that, we'll go back to the physiological functions of the heart and describe them in more details. The blood vessels include the aorta and the large branches of the aorta, the large arteries, and these function as distributing channels. They distribute oxygenated blood and nutrients to the various tissues of the body, and because of that we need to have high pressure in order to push blood along the systemic circulation and distribute the oxygenated blood and nutrients to the tissues. We need to have high pressure in the aorta and in the large arteries. The small arteries and the arterioles, specifically the arterioles, they function as the resistance blood vessels, and the highest resistance to the blood flow in the circulation is met at the level of the arterioles, and this is important. And it is because of this high resistance there is marked drop in the blood pressure at the level of the arterioles. This is physiologically important so that the capillaries are not exposed to high pressure. The capillaries are the parts of the circulatory system where we have the exchange. Then we have the venules and the veins, which are the capacitance vessels. They can accommodate large volume of blood because the walls of the veins are highly distensible. They are highly compliant. In this diagram, we have the major components of the cardiovascular system again. The heart functions as the central pump of the cardiovascular system. The arteries as distributing channels. Here we have the arterioles. They are the resistance blood vessels. The capillaries, the exchange region. And the veins and the venules, the capacity vessels. The arteries have thicker walls than veins of the corresponding size, but they have lower compliance than the veins. The walls of the veins are highly distensible, and because of that, the veins have high compliance. They can accommodate large volume of blood, so they function as volume reservoirs. In fact, approximately 2/3 of the blood volume is present in the veins. On the other hand, the arteries have much higher pressure than the veins in order that they can push blood along the circulation. And because of that, the arteries are considered to be pressure reservoirs. So now we have finished with the brief description of the functions of the heart as the central pump of the cardiovascular system and the functions of the different blood vessels. And now let's describe the functions of the heart in more details. Inside the cardiovascular system, blood moves along two circulations, the systemic and the pulmonary circulations. The left ventricle is considered as the pump of the systemic circulation, and the right ventricle is considered as the pump of the pulmonary circulation. These two circulations are connected in series, and this means that one circulation ends and the other circulation begins. The systemic circulation begins in the left ventricle, where oxygenated blood is ejected into the aorta. The aorta and the large branches of the aorta distribute the oxygenated blood and nutrients to the tissues. The capillaries, where we have exchange, then the venules and the vein, they collect deoxygenated blood and return it to the right atrium. And it is here where we have the end of the systemic circulation. Then the pulmonary circulation begins in the left ventricle, where the oxygenated blood is ejected into the pulmonary trunk. The oxygenated blood flows through the pulmonary arteries, pulmonary capillaries, to get reoxygenated. Then the oxygenated blood returns to the left atrium via the four pulmonary veins, and it is in the left atrium where the the pulmonary circulation ends. In this diagram, this is the left ventricle ejecting oxygenated blood into the aorta. These are the systemic arteries, the systemic arterioles, the systemic capillaries, the systemic venules, the systemic veins, which belong to the systemic circulation. And it is again in the right atrium where we have the end of the systemic circulation. Then we have the beginning of the pulmonary circulation in the right ventricle. Deoxygenated blood is pumped into the pulmonary trunk, the pulmonary arteries. Blood flows through the pulmonary capillaries so that the deoxygenated blood is recharged again with oxygen. Oxygenated blood returns to the left atrium via the pulmonary veins. And it is in the left atrium where we have the end of the pulmonary circulation. Then the systemic circulation begins again in the left ventricle. So it is evident from this diagram that the two circulations, the systemic and the pulmonary circulations are connected in series. So the heart functions as the central pump of the cardiovascular system. If the heart of an experimental animal, like the mouse or the rat or the rabbit, is excised and placed in a beaker containing solution with appropriate concentrations of glucose, calcium, sodium, potassium, the heart continues to beat. This experiment shows that the stimulus which stimulates the heart to beat originates from the heart itself. This is because when we excise the heart from the experimental animal, we cut all the sympathetic and the parasympathetic nerves which are connected to the heart. Now we are placing the heart in the solution, and the heart continues to beat. So this shows that the stimuli which stimulate the heart to continue beating originate from within the heart itself. And this is the function of a specialized system in the heart composed of modified cardiac muscle cells. This system has the functions of, number one, to generate the cardiac stimuli, which we call them the cardiac impulses, and number two, to propagate, to conduct these cardiac impulses so that these cardiac impulses reach the cardiac muscle cells which we have in the walls of the atria and the ventricles and stimulate them to contract. This specialized conduction system of the heart is composed of the impulse generator, the sinoatrial node, where the impulses are generated, a delaying device, the atrioventricular node, where we have delay in the conduction of the impulse, and this delay is known as AV nodal delay, and we'll describe the physiological significance of this AV nodal delay in the subsequent lectures. Then we have the AV bundle, the atrioventricular bundle, the bundle of Hess, the right bundle branch, and the left bundle branch, and the Purkinje fibers, which are the fine terminal branches of the left and right bundle branches. It is thus evident that the heart has four basic physiological properties which enable it to function as the central pump of the cardiovascular system. These physiological properties are autorhythmicity, which is the ability of the heart to generate its own stimuli in a regular rhythmical way, conductivity, which is the ability of the heart to conduct these stimuli so that the cardiac stimuli reach the cardiac muscle cells which we have in the walls of the atria and the ventricles. And when these stimuli reach the cardiac muscle cells in the walls of the atria and the ventricles, they excite them. So excitability is the third important physiological property of the heart. And when these cells are excited, they respond electrically and mechanically by contraction. So contractility is the fourth basic physiological property of the heart. So within the heart, then, we have two main types of cells. The cells of the specialized conduction system in the sinoatrial node, AV bundle, bundle branches, and Purkinje fibers, and the contractile cells which we have in the walls of the atria, interatrial septum, and the walls of the ventricles and interventricular septum. These contractile cells are also known as the working cells or the working fibers or the myocytes. Now let's describe the ultrastructure of a contractile cardiac muscle cell, the working cell or the myocyte. The cardiac muscle cells or the fibers or the myocytes, they are cylindrical cells with centrally located nuclei. The cells branch, as you can see, this cell, this one branch of the cell, and this is another branch. This is another cell, this is branch, and this is another branch of the cell, and they interdigitate. But each cell is a complete unit surrounded by a plasma membrane, which is also known as the sarcolemma. Where the end of one cardiac muscle cell abuts on another cardiac muscle cell, so this is one cardiac muscle cell abutting on another cardiac muscle cell, the membranes of both fibers parallel each other through an extensive series of folds, and these areas where we have the extensive series of folds are known as the intercalated discs. So this is an intercalated disc. 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 And this is the sarcolemma of this fiber, and this is the sarcolemma of the other cardiac muscle cell or fiber. Thus, each cardiac muscle fiber is separated from its neighboring fibers by its sarcolemma laterally and by the intercalated discs end to end. The intercalated discs are important because they enable the synchronization of the contraction of the cardiac muscle fibers by providing both mechanical and electrical coupling. So how the intercalated discs provide mechanical and electrical coupling of the cardiac muscle cells. This is the cardiac muscle fiber, this is another fiber, this is an intercalated disc, this is a cardiac muscle fiber, this is another fiber which branches, this is another fiber, this is another fiber, and these are intercalated discs. This is another intercalated disc, and let's take this intercalated disc and magnify it and see what do we have in the intercalated discs. We have desmosomes, and these desmosomes are the structures which provide the mechanical coupling of the cardiac muscle cells, because the desmosomes, they resist stretching and prevent detachment of the cardiac muscle fibers during contraction. So they ensure that the cardiac muscle, the contractions of the cardiac muscle fibers are synchronized. In the intercalated discs, we also have gap junctions, and it is these gap junctions which provide electrical coupling of the cardiac muscle cells. So the gap junctions are transmembrane channel proteins connecting the cytoplasm of the neighboring cardiac muscle cells. They allow free diffusion of ions between the neighboring cardiac muscle cells, so the action potentials can also travel from one cardiac muscle cell to another cardiac muscle cell. Accordingly, the intercalated discs allow the passage of current through the cardiac muscle cells, and this mechanism ensures electrical coupling of the cells, so the cardiac muscle cells are excited at the same time, and they respond by contraction also at the same time. So this ensures the synchronization of the contractions of the cardiac muscle fibers. It is because of this synchronization of the cardiac muscle fibers, the synchronization of the contractions of the cardiac muscle fibers, and the mechanical and the electrical coupling of the cardiac muscle cells via the intercalated discs, cardiac muscle functions as a syncytium. So cardiac muscle functions as a syncytium, as each cardiac muscle cell is electrically connected to its neighboring cells through the gap junctions. Thus, when a single cardiac muscle cell, when a single myocyte is stimulated, it responds electrically by the generation of a fast action potential in the sarcolemma of the myocyte. This action potential spreads from one cardiac muscle cell to another cardiac muscle cell through the gap junction. Thus, contractions of the cardiac muscle cells occur at the same time because the cells are excited at the same time. So we have synchronous contraction of all the myocytes. In the heart, we have two syncytia, the atrial syncytium and the ventricular syncytium, and these two syncytia are separated by the fibrous tissue which surrounds the valvular openings, which we call it the fibrous skeleton of the heart. This is again a cardiac muscle cell, a contractile cardiac muscle cell, a mechanical cell, a working cell, or a myocyte. And at the ultrastructural level, the cardiac muscle cells, the myocytes, they typically resemble the skeletal muscle cells or skeletal muscle fibers in also having myofibrils, which are packed in the sarcoplasm. The myofibrils are composed of sarcomeres. Also, we have specialized transverse tubular and sarcoplasmic reticular systems. We have the T-tubules and the membranes of the sarcoplasmic reticulum. However, the T-tubules, the T-tubular system in the cardiac muscle cells is located at the Z lines of the sarcomeres rather than at the AI junctions in the sarcomeres of the skeletal muscle fibers. The sarcoplasmic reticulum makes complexes with the transverse tubular membrane at dyads rather than triad junctions, which we have in the skeletal muscle fibers. The cardiac muscle cells contain large amount of mitochondria, and this is very important in order to provide continuous supply of adenosine triphosphate for the cells. And they also have rich capillary supply, about 1 capillary per fiber. It is within the sarcoplasmic reticular membrane systems where we have calcium ions stored inside the myocytes. These calcium ions should be released from the sarcoplasmic reticulum in order that the myofibrils contract. In myocytes, a significant calcium influx comes from the extracellular space during the plateau phase of the action potential. The phases of the action potential will be described in details in the next physiology lecture in the cardiovascular block. This is different from what we have in the skeletal muscles. We do not have calcium influx during the action potential in skeletal muscle fibers. But this influx of calcium in the myocytes is important. The role of calcium ions in the excitation contraction coupling in myocytes is similar to their rules in the skeletal muscle fibers. And as you all know, the term excitation contraction coupling means the mechanism by which the action potential causes the myofibrils, the sarcomeres of the myofibrils of the muscle cell to contract. However, in the cardiac muscle cells, in the myocytes, it is the influx of extracellular calcium through voltage-sensitive dihydropyridine receptors in the tubular system, which triggers calcium-induced calcium release through the ryanodine receptors at the sarcoplasmic reticulum. So in the T system, in the transverse tubular system, we have voltage-gated calcium channels, which we call them L-type calcium channels. We also call these L-type calcium channels dihydropyridine receptors, because dihydropyridine can bind to these receptors and can block these calcium channels. In the membranes of the sarcoplasmic reticulum, we have calcium channels, which are also known as ryanodine receptors, because the ryanodine, which is a chemical substance, can bind to these receptors. The ryanodine receptors which we have in the sarcoplasmic reticulum of the cardiac muscle cells are different from the ryanodine receptors which we have in the skeletal muscle cells. The release of calcium from the sarcoplasmic reticulum of the cardiac muscle cells is induced by the influx of calcium through the voltage-gated L-type calcium channels in the transverse tubular system and in the sarcolemma. And this mechanism is known as calcium-induced calcium release. So the release of calcium from the ryanodine receptors at the sarcoplasmic reticulum is induced by calcium ions getting in from the extracellular space through the dihydropyridine receptors, through the L-type voltage-gated calcium channels which we have in the transverse tubular system and in the sarcolemma as well. So here we have a healthy cardiac muscle cell, a healthy cardiac myocyte. This is a transverse tubule, and this is the sarcolemma. This is the sarcoplasmic reticulum. These are the myofibrils, which are composed of myofilaments. This is an L-type calcium channel, a voltage-gated L-type calcium channel, which is also known as dihydropyridine receptor. In the sarcoplasmic reticulum, we have ryanodine receptors, which are also calcium, function as calcium channels. So when a myocyte is excited by a cardiac stimulus, by a cardiac impulse originating from the sinoatrial node and reaching the myocyte through the specialized conduction system of the heart, the myocyte responds electrically by generating a fast action potential in the sarcolemma. So this is now a fast action potential generated in the sarcolemma. This fast action potential propagates along the sarcolemma and travels down the transverse tubular system to the fibrous interior. And when the action potential reaches the T-tubular system and the voltage is slightly changed, the voltage-gated L-type calcium channels, the dihydropyridine receptors, are activated and opened by the action potential, and thus calcium ions get into the cell. So we have an influx of calcium from the extracellular space into the myocytes. This external calcium influx is required for the excitation contraction coupling in the heart, but this influx of calcium ions itself is not sufficient to produce contraction. Rather, it is used to trigger the release of calcium from the sarcoplasmic reticulum. How this happens? So, the calcium ions coming into the cell through the L-type voltage-gated calcium channels bind to ryanodine receptors in the sarcoplasmic reticulum. And these ryanodine receptors, again, they function as calcium channels for the calcium ions which are stored here in the sarcoplasmic reticulum. So this calcium ion acts as a ligand for the ryanodine receptor in the sarcoplasmic reticulum. So the voltage-gated calcium channels, the L-type voltage-gated calcium channels, the dihydropyridine receptors, they are usually very near to the ryanodine receptors, so that calcium influx will induce high local levels of calcium ions. At micromolar calcium levels, these are the local levels of calcium ions, the ryanodine receptors, now these calcium ions, they function as ligands for the ryanodine receptors, and the ryanodine receptors open, releasing stored calcium from the sarcoplasmic reticulum. Then this calcium binds to troponin C, and this initiates the sliding of the thin filaments past the thick filaments, as we will describe in the subsequent slides. This calcium-induced calcium release from the sarcoplasmic reticulum is pH sensitive. It shows strong inhibition when the pH inside the myocyte is 6.5, and it shows maximal release at physiological pH of 7.4. So here we have again a summary of the mechanism of calcium-induced calcium release through the ryanodine receptors at the sarcoplasmic reticulum. So these are the L-type calcium channels, the dihydropyridine receptors, which we have in the T-tubular system, and we also have them in the sarcolemma. And these are the ryanodine receptors, which we have in the sarcoplasmic reticular membranes. Now let's very briefly describe the steps of the excitation contraction coupling in the myocytes. The excitation contraction coupling is similar in cardiac and skeletal muscles. The contraction is triggered by an increase in the intracellular concentration of calcium ions, which bind to troponin see on the thin filaments of the myofibrils. However, in the cardiac muscle cells, we mentioned the release of calcium from the sarcoplasmic reticulum is induced by the influx of calcium from the extracellular space. So tension generation in cardiac muscle cells, but not in skeletal muscle cells, is profoundly influenced by the extracellular calcium levels and also by the factors which influence the magnitude of the inward calcium current. For example, if the concentration of extracellular calcium ions is doubled in the extracellular fluid, this may nearly double the maximum cardiac contractile force, because the more calcium we have in the extracellular fluid, the more calcium gets in, the more calcium is released from the sarcoplasmic reticulum, and the more calcium is available for the contraction. Thus, drugs which reduce the influx of calcium through the voltage-gated L-type calcium channels have profound negative inotropic effects on the myocardium. They decrease myocardial contractility. Inotropic effects, these are the effects on contractility, and we will describe what's the meaning of inotropic effects in the subsequent slides. These drugs will reduce the calcium influx and thus will have profound influence, profound negative effects on the contractility in cardiac but not in skeletal muscles. They would affect skeletal muscle contractility only when they are present in massive doses. So now let's describe the mechanism of sliding of the thin filaments past the thick filaments in the sarcomeres of the skeletal muscle, of the cardiac muscle cells. So here we have a cardiac muscle fiber, a myocardial fiber, or a myocardial cell, or a myocyte. And as we mentioned, the sarcoplasm, the cytoplasm of the fiber is packed with myofibrils. The myofibrils are composed of sarcomeres, and the sarcomeres are composed of myofilaments. The thick myofilaments are known as myosin filaments, and the thin myofilaments are known as actin myofilaments. So here we have a sarcomere in the relaxed state, and here we have a sarcomere in the contracted state, where the thin filaments, the thin myofilaments, slide past over the thick myosin filaments. The thick filaments are composed of myosin molecules. Each myosin molecule has got one tail and two globular heads. The myosin molecules combine together to form a thick filament, which we call it myosin filament. So the myosin filaments, or the thick filaments, are composed entirely of myosin molecules. Now, a key point important in the sliding filament mechanism is that the heads of each myosin molecule have an actin binding site. This is a site which binds to the actin molecules in the actin myofilaments, and also an ATPase site which splits adenosine triphosphate. And this splitting of the adenosine triphosphate is important in the contraction cycle, as we will see in the subsequent slides. These are the thin filaments. And in the thin or actin filaments, we have three types of proteins. We have actin molecules, and in these actin molecules, on these actin molecules, we have binding sites for the globular heads of myosin. However, in the relaxed state, the myosin binding sites on the actin molecules are covered by another protein in the thin filaments known as thromboyosin. A third type of protein which we have in the thin filaments is troponin. So when the myocytes are excited and we have calcium-induced calcium release from the sarcoplasmic reticulum, and we have a rise in the intracellular concentration of calcium ions, calcium binds to troponin C, and when this happens, calcium troponin C complex interacts with thromboyosin, and this results in uncovering of the myosin binding sites on the actin molecules, and now the binding sites on the actin molecules are ready for the myosin heads to bind to. So in this diagram we have the steps of the contraction cycle. So these are the globular heads of the myosin molecule, and this is the tail of the myosin molecule. And the globular heads of the myosin molecule are attached to their binding sites on an actin molecule. Now, these heads of the myosin molecule, they need to be detached from the actin molecule so that they can bind to another actin molecule, and the contraction cycle is repeated. How this happens? ATP binds to myosin head, and this binding causes the dissociation of the actin myosin complex. So now the globular heads of myosin are detached from the actin molecule. Then adenosine triphosphate is hydrolyzed into adenosine diphosphate and inorganic phosphate, and this hydrolysis of adenosine triphosphate returns the globular heads of myosin to their resting conformation. These heads, they need to return to their resting conformation so that they can bind to another actin molecule. They can bind to their binding site on another actin molecule. Then a cross bridge forms, and the myosin head binds to a new position on the actin filament, to a new molecule in the actin in the thin filaments. Phosphate, the inorganic phosphate, is released, and when the inorganic phosphate is released, the myosin heads change their conformation. Look, this is the resting conformation. Now this is the conformation when the inorganic phosphate is released. Now when the myosin heads change their conformation, this results in the power stroke, and the filaments slide past each other. Then lastly, adenosine diphosphate is released, and the contraction cycle and the power stroke cycle is repeated again. So by definition, contraction is the continuous cycling of cross bridges between the globular heads of myosin and their binding sites on the actin molecules. And as long as calcium contractions remain high, actin and myosin cross bridges interact to produce contraction of the sarcomeres, and this contraction is similar to the contraction of the sarcomeres, which we see in skeletal muscle fibers. This is a nice animation which shows the contraction cycle, and it is important to note that there is a major difference between the skeletal muscle fibers and the cardiac muscle fibers as to their susceptibility to rigor. Skeletal muscle cells are susceptible to rigor if no adenosine triphosphate is present in the muscle cell, because it is the binding of the adenosine triphosphate to the myosin heads which detach the myosin heads from the actin filaments, so that the cycle is repeated again. So when adenosine triphosphate is not present, the myosin globular heads are not detached from their binding sites on the actin molecules, and the fiber undergoes rigor. Cardiac muscle cells are unlikely to ever undergo rigor because they have large amounts of mitochondria which produce continuous supply of adenosine triphosphate. So now calcium is important for the contraction cycle. Calcium is important for the contraction. What about relaxation? How the cardiac muscle fibers undergo relaxation? We need to have low intracellular calcium ions between successive action potentials. And we do have mechanisms which maintain low intracellular calcium ions between successive action potentials. One of these important mechanisms is the presence of calcium pumps, calcium ATPase pumps in the plasma membrane and in the sarcoplasmic reticular membranes. So the plasma membrane, the sarcolemma, and the sarcoplasmic reticular membranes, they contain calcium ATPase pumps which translocate calcium ions back into both the extracellular fluid and the leumina of the sarcoplasmic reticulum. And these are ATPase pumps because the calcium ions are transported against their concentration gradient. When calcium ions are released from the sarcoplasmic reticulum as a result of the calcium-induced calcium release mechanism, this doesn't mean that the concentration of calcium ions now in the sarcoplasmic reticulum is low. Still, the concentration of calcium ions in the sarcoplasmic reticulum is high. Also, the concentration of calcium ions in the extracellular space is high. So when calcium ions are returned back to the extracellular space and to the sarcoplasmic reticulum, calcium ions are pumped. They need to be transported against their concentration gradients. The activity of the sarcoplasmic reticulum pump the calcium ATPase, the SIRK, K means calcium, is modified by a protein known as phospholamban. So this is the calcium ATPase pump in the sarcoplasmic reticulum, and the activity of this pump is influenced by a protein known as phospholamban. Phospholamban speeds up the pump when it is phosphorylated by cyclic adenosine monophosphate protein kinase. So catecholamines, norepinephrine and epinephrine, noradrenaline and adrenaline, when they bind to their adrenergic receptors in the sarcolemma, they activate adenylyl cyclase in the sarcolemma, an enzyme known as adenylyl cyclase, resulting in an increase in the intracellular concentration of cyclic adenosine monophosphate, which functions as a second messenger for norepinephrine and epinephrine. And this activates protein kinase, which phosphorylates phospholamban, resulting in speeding up the removal of the calcium ions. It is also important to note that norepinephrine and epinephrine, when they bind to their adrenergic receptors and stimulating adenylyl cyclase and have cyclic adenosine monophosphate acting as their second messenger and stimulating the protein kinase enzyme, this also results in phosphorylation of the L-type voltage-gated calcium channels. So we have more influx of calcium. So catecholamines, they increase the force of contraction, and they also speed up the removal of calcium from the sarcoplasmic reticulum by inducing phosphorylation of phospholamban. So it is very beautiful. These catecholamines, they increase the force of contraction because they phosphorylate the L-type calcium channels, having more calcium influx, having more release of calcium from the sarcoplasmic reticulum, and they also have an influence in phospholamban speeding up the removal of calcium. So they result in stronger contraction and also speeding up the relaxation process. Another mechanism for maintaining low intracellular calcium between successive action potentials is the presence of a sodium calcium exchanger, and this exchanger is present only in the plasma membrane. So the sarcolemma also contains an exchanger, a sodium calcium exchanger, which drives calcium efflux across the plasma membrane. So the exchanger removes one calcium ion from the cytoplasm into the extracellular space for every 3 sodium ions which enter the cell down their electrochemical gradient. So we need to have a low intracellular concentration of sodium so that sodium ions get in through the sodium calcium exchanger, and when 3 sodium ions get in through the sodium calcium exchanger, 1 calcium ion is removed from the cytoplasm back into the extracellular space. What maintains low intracellular concentration of sodium ions within the cytoplasm of the myocyte is the sodium-potassium pump. So the exchanger then, the sodium-calcium, the calcium-sodium or the sodium-calcium exchanger utilizes the energy from the influx of sodium ions down their electrochemical gradient. Previously, this gradient is previously established by the ATPase coupled sodium-potassium pump. So, increases in intracellular sodium concentration, if the concentration of sodium ions in the intracellular space in the cytoplasm increases, this would decrease the inward electrochemical gradient, which drives sodium ions entry, and this results then in an increased internal calcium concentration. How? If there is high sodium ions inside the cell, inside the myocytes, this calcium sodium exchanger will work in an opposite way, will work in a reverse way. It will work to bring one calcium ion from the extracellular fluid into the cell for every 3 sodium ions moved out of the cell. And this is in fact the mechanism of action of digitalis or digoxin, and these are drugs which are used in the management of cardiac failure. These agents, they block the sodium-potassium pump. So sodium ions accumulate in the cytoplasm of the myocytes. And when the sodium ions accumulate in the cytoplasm of the myocytes, the electrochemical gradient for sodium is decreased, thereby allowing an increase in the intracellular sodium concentration And when this happens, the sodium-calcium exchanger acts in a reverse direction, as we have just mentioned. So this diagram shows the sodium-calcium exchanger in the sarcolemma. So for every 3 sodium ions getting in down their electrochemical gradient, we have 1 calcium ion gets out. Now these sodium ions, they get in down their electrochemical gradient because the concentration of sodium ions is low in the cytoplasm. And what keeps this low intracellular concentration of sodium is the sodium-potassium pump. So if this pump is inhibited, the concentration of sodium increases in the intracellular fluid in the cytoplasm, and the exchanger works in a reversed way. So at the end of this lecture, let's very briefly describe the factors which affect cardiac contractility. Cardiac contractility is in fact an intrinsic property of the contractile cardiac muscle cells, and it is defined as the force of contraction of the heart. And this is, as we mentioned earlier, this is essential for effective pumping of the heart and for the heart to function as the central pump of the cardiovascular system. In physiological terms, cardiac contractility is referred to as the inotropic state of the myocardium. So when we say inotropic state of the myocardium, we mean the inotropic, the cardiac contractility. An inotropic effect is an effect or a mechanism which thus affects cardiac contractility. Positive inotropic effects are the effects or the factors and the mechanisms which increase the force of contraction, which increase cardiac contractility. For example, sympathetic stimulation and the release of catecholamines, norepinephrine and epinephrine, and an increase in the calcium ions in the sarcoplasm of the myocytes, they all have positive inotropic effects. On the other hand, negative inotropic effects are the effects or the factors or the mechanisms which decrease the cardiac contractility. For example, parasympathetic stimulation, the vagal stimulation and release of acetylcholine, they decrease the cardiac contractility of the atria. So the catecholamines, the sympathetic stimulation, when the sympathetic system is stimulated, we have release of catecholamines, epinephrine and norepinephrine, and these interact with beta-adrenergic receptors in the sarcolemma, and when this happens, they activate an enzyme known as adenylyl cyclase in the sarcolemma, and this increases the intracellular levels of cyclic adenosine monophosphate. The cyclic adenosine monophosphate activates protein kinases which promote phosphorylation of the voltage-gated L-type calcium channels which we have in the T-tubular system in the sarcolemma. These are the dihydropyridine receptors, so we have more influx of calcium. And the more influx of calcium we have, the more release of calcium from the sarcoplasmic reticulum we have, and the more calcium is available for the contraction cycle, so we have stronger contraction. So sympathetic stimulation thus results in a positive inotropic effect. We mentioned also earlier that sympathetic stimulation, catecholamines, also induce phosphorylation of phospholamban, and this speeds up the removal of the calcium ions from the cytoplasm back into the sarcoplasmic reticulum, and hence speeding up relaxation. So it is very beautiful again that catecholamines, they not only increase the force of contraction, not only they have a positive inotropic effect, but they also speed the relaxation during diastole. Parasympathetic stimulation, the mechanisms of parasympathetic influences on cardiac contractility involve the interaction of acetylcholine with muscarinic receptors on the cardiac muscle cells, And also, this is the vagus releasing acetylcholine, acetylcholine interacting with muscarinic receptors on the cardiac muscle cells. Also here we have the vagus releasing acetylcholine, and this acetylcholine inhibits the release of norepinephrine from the neighboring sympathetic neurons. When acetylcholine binds to its muscarinic receptors on the cardiac muscle cells, this interaction inhibits the enzyme adenylyl cyclase, resulting in a decrease in the intracellular levels of cyclic adenosine monophosphate, and the reduction in the cyclic adenosine monophosphate levels leads to a reduction in the calcium influx during the action potential, and thus a decrease in contractility. It is very important to note that the reduction in contractility induced by parasympathetic stimulation is seen primarily in the atria and not in the ventricles. And that's the end of this lecture. And here we have the active learning objectives, the intended learning outcomes again. And thank you very much for listening.